Good morning to everyone. Today is the Graduate Research Day. It's the day we celebrate the work you have done over the year. And I would like to start this activity by thanking you for the hard work you do and be enthusiastic about all the excellent activity that we will have today over this research day. We have a very tight program, so I will immediately start with the first activity that we have for today, which is the keynote speaker. I'm very honored and delighted to introduce Dr. Reinhart-King. She's a Cornell Vanderbilt professor at the engineering and the director of the graduate studies in the Department of Biomedical Engineering at Vanderbilt University. She was a faculty before 2017. She was a faculty at Cornell University. She obtained undergraduate degrees in chemical engineering and biology at MIT, and her PhD work was at University of Pennsylvania in the Department of Engineering. Her focus is in cell mechanics and cell migration with application to cancer, arteriosclerosis. She has many prestigious awards that you can see in the flyer that was circulated. I want to say she's a standing member of one of the NIH study section and is chair of the Diversity and Inclusion Committee of AI MBE. So with no more, I would like to thanks first Dr. Reinhart-King for being here on a busy day, because today is the recruitment day for their department. And without more, I will let you enjoy her talk.
Perfect. Let me just share my screen here. All right, well, thank you for the introduction, Dr. Derriera, and thank you, Dr. Hutchison, for inviting me. And I'm happy to be here. I'm sorry I can't join you in person or share your whole day, but I will say I love the intro because it started by saying celebrating your research in the past year. And I was actually just on a panel this week. Somebody asked me, what's the key to surviving graduate school or research? And my answer was celebrating the successes. So certainly enjoy your day and remember to celebrate all the research successes you've had in the past year. So with that, I'm going to talk about some work my lab's been doing. We work in the area of mechanobiology and mostly related to cancer. And so I'll talk a little bit about that. And really our goal is bringing what we're finding in mechanobiology into medicine. So trying to identify therapeutic targets that are actually viable clinically. So with that, this is, I always show this slide. This is sort of our approach to basically how we tackle this problem, particularly as biomedical engineers. Because you think about a lot of different strategies when you think about disease. And I think one of the things that engineers do well is physical and chemical characterization and then model building. And so how do we incorporate that into our research? And so really, we generally start here with the human pathophysiology. So we're inspired a lot by human disease and human tissue, looking at patient samples or clinical images and trying to understand what disease actually looks like. Then we do a lot of ex vivo characterization. So again, as engineers, our toolbox is really physical and chemical measurements. So things like looking at mechanical properties, using things like atomic force microscopy and compression testing, doing a lot of imaging, whether it's through histology or optical imaging or even non-optical approaches, trying to understand what the fiber architecture of the tissue looks like, the cell organization of the tissue, the mechanical properties, with the idea that once we have that information, we can basically use all of the same tools that tissue engineers are using in biomaterials and microfabrication, different types of chemistries and things like this to basically build in vitro models of the characteristics of the disease tissues that we've defined. So it's basically tissue engineering of disease with the goal of trying to understand how the cells behave in those disease environments. So the benefit of engineering these systems is that we can then use them like dials to tune them to understand, for instance, what a change in mechanics or a change in fiber structure or change in fiber architecture is going to do in terms of affecting disease progression. With the idea of once we identified the cellular properties, things like proliferation and migration and adhesion and just fundamental properties of cell behavior, we can then go in and understand the molecules that are controlling these behaviors. And then this is where the translation comes in, is really trying to understand which are the molecules that are most important in these processes, because those become the therapeutic targets. So we bring to bear basically a molecular biology toolbox, genomics and proteomics, and your standard molecular biology tools to manipulate those molecules to ultimately intervene. And then I add in, usually we're intervening in mouse models or ex vivo chick models, not typically straight back into humans. So with that, my lab's really interested in 3 problems: atherosclerosis, metastasis, and angiogenesis. Unfortunately, I never have time to discuss all of them. So I'm really going to tell you the first part of my talk is going to be a recent story we've been working on related to metastatic migration. And then the second part will be some work that we've been doing in angiogenesis. Okay, so metastasis, I'm sure most of you are familiar with the idea that basically cancer, of course, the reason most individuals die from cancer is not usually the primary tumor, it's when it spreads to secondary sites. And the way that happens is cells typically detach from the primary tumor, they migrate out into the surrounding tissue, they enter either the lymph system or the circulatory system, and then travel off into secondary sites. And so it's really this stage here that my lab's interested in. So how do cells get from the primary tumor out into the circulatory system? What are the mechanisms cells use to migrate? So we've done a lot of work in the space. I'm going to tell you just about basically one story we've been working on that's more recent in our lab. And it was really inspired by these papers that Hanahan and Weinberg have published. If you're not familiar with them, this paper that came out in Cell in 2000, I think is the most highly downloaded paper ever published in Cell. So if you're interested in cancer, it's definitely the one that you should read, one of many, I suppose. And basically what they tried to do is identify different aspects of cancer that basically define a tumor. What defines a cancer cell? What defines a tumor? Things like sustained proliferation, evading apoptosis, activating an invasion and metastasis, inducing angiogenesis. So these have really become sort of the staples of the cancer field of the aspects that people have been focusing on. It was really a good summary. But then about 11 years after that, 2011, Hanahan and Weinberg came out with an updated version of emerging hallmarks, and they identified deregulation of cellular energetics as one of them. You might recognize some of the others, so avoiding immune detection. This comes to the idea of immunoengineering and is basically a good summary of basically where the field is going in terms of immunoengineering, cancer immunoengineering. Tumor inflammation, so there's significant information, significant data coming out about things like macrophages and inflammatory cell infiltration into tumors, things like this. But this was the one that sort of sparked our interest, this deregulation of cellular energetics. And it's a pretty old field. So you've probably heard of things like the Warburg effect and the reverse Warburg effect, the idea that cancer cells need and utilize energy differently than healthy cells. But we wanted to put it in the context of the problems that we had been thinking about, which is cell migration. And so when you look at these hallmarks of cancer that Hanahan and Weinberg had defined, so these are the original ones, and then you just put it through the lens of metabolism, they're actually incredibly intertwined. So metabolism plays into almost all of these things. The ability to proliferate requires energy. The ability to undergo angiogenesis, for instance, is affected by things like oxygen deprivation, which influences metabolism. Migration is affected by the acidic environment of the environment, as well as oxygen deprivation. So really, it's almost impossible to separate the hallmarks of cancer from metabolism, because in fact, they're almost all in part mediated by it. And so when you think about metabolism, how's energy actually used in a cell? If you ask most people in the cancer community, they will tell you that the reason they care about metabolism and the reason they care about energy utilization is because of proliferation. So it takes a whole lot of energy for the cell to basically remake all of its proteins, to duplicate its chromosomes, and to divide. That's a very energy intensive process. And so to do so, they have to upregulate a number of metabolic pathways. If you look at individual tissues, there's also things like making energy and storing it, gluconeogenesis, action potentials, and neurons require a lot of energy, not surprisingly. Establishing gradients across the cells to allow for proton leak and non-mitochondrial energy production take a lot of energy. The one that really intrigued us was actin-myosin activity. So again, as engineers, one of the things that we thought about, sure, it takes a lot of energy to proliferate, but what about to migrate? So what about to actually move? How much energy does that take? And in terms of what was known in the field about this, there were a few hints. So there was this work that came out of the neuroscience field. So these are neurons, where basically what they did was they took the cells and they blocked their ability to make ATP. So the cells have the ATP that they have, but they can't make any more of it. And then they just look to see how quickly the ATP depletes itself. So if the cell can't make more, how quickly does the cell run out of ATP? And the top line here is the control situation. So after blocking ATP synthesis, within just a few minutes, a cell depletes all of its energy. But if you take the cell, inhibit various things associated with the actin cytoskeleton, so you basically inhibit the ability of a cell to contract, you eliminate the ability of a cell to build this architecture, the cytoskeleton that makes up the cell. What happens is you get significantly less ATP depletion. So the cell is continuing to store some amount of its ATP if you prevent cytoskeletal activity. And so this was really our first hint that the cytoskeleton is really a key player in generating and using ATP. And so this was what we launched from, saying, well, if the cytoskeleton has a key role in storing and creating ATP, then potentially it also has a key role in migration. And so this was our question, whether or not the ability of a cell to migrate is related to how much energy it has. And how much energy does it take for a cell to move? And as a cell moves, it has to move itself, right? So it needs the energy to move itself, but also has to reorganize the tissue it's moving through. So it has to wind its way through the tissue, it degrades the matrix, it contracts the matrix, all of this to navigate through. And we wanted to know basically how much energy does it take? It doesn't matter. So, again, if you asked somebody this question 5 years ago, they would have said, well, it takes energy, but not nearly as much energy as it takes to proliferate. So let's just focus on proliferation. Okay, so these were our questions. We wanted to know about the relationship between migration and energy utilization. In other words, if a cell has more energy, is it better at migrating? How does the terrain affect the energetic needs during migration? So if you make the environment that the cell has to get through harder or stiffer or less porous, basically a tougher environment to move through, does it require more energy? Whether or not the energetic needs affect migration decisions. So in other words, if you were to give a cell a choice between a high energy path and a low energy path, is it going to be smart enough to choose the path that takes less energy? And then lastly, what the role of energetics is in collective migration. So we'll talk about that and specifically what collective migration is and why we care about it in the context of energetics. All right, so my lab has been focused on migration for quite some time. And one of the things that we've been interested in with regard to migration is how the environment that the cells move through affects their ability to move. And so this is actually an ex vivo, it's an explant of a tumor. So it's basically a mammary tumor we took out, put onto a microscope stage. And the purple is the collagen extracellular matrix. So it's the network that the cells have to move through. and the green are the cells. And so basically what you see here is over the same time window, you can find cells that zip right through. See this cell squeezing up through this pore and across the screen. And in the same time frame, these cells aren't really going anywhere at all. And so the question is, there's something about these cells or this environment that's conducive to the cells moving. Even if you just take cells and put them on a petri dish, what you can find is cells that zip across the screen, very motile cells, and you can find other cells that don't really do anything in the same time. A lot of heterogeneity in cell behaviors. And so we've been asking for quite some time whether or not the behavior of cells is actually determined by their environment or determined by inherent characteristics of the cells. So is it stochastic? So is it changing in time? Or is it deterministic, meaning these cells were always meant to be high migrators? And whether or not the ability of the cells to migrate is dependent on the environment they're moving in. So in other words, can you give them a certain matrix that's going to induce an otherwise non-motile cell to get up and go? Okay, so with relationship to the problem that we're talking about here, one of the things that we've been focused on in setting up these experiments is trying to create environments that are as in vivo-like as possible. So the experiments that we do would be hard to do in vivo. Intravital microscopy in real time of cell migration is very difficult. And so how can we create a platform in vitro that allows us to do that? If you just buy collagen, so you go to Becton Dickinson or Sigma and order some collagen and you polymerize it like they instruct you to, it basically looks like this. So it's a bunch of fibers. And a good part of the literature basically says, well, this looks pretty close to this. So at least we've got a fibrous architecture and we've got pores. But one of the things that we've noted is that this is hardly a homogeneous environment, right? You've got big pores and small pores. You've got areas of alignment. You even have tunnels that move through this stromal ECM. And so one of the things that my lab's been doing is basically working out protocols to make denser collagen matrices or looser collagen matrices with larger fibers to even align the collagen matrix into these tracks and even to micromold the collagen so that you can get these tunnels that the cells are able to move through. So I'm sort of laying the foundation for what the platform is that we're going to use to ask these questions about energy. So we took these platforms and then said, how much energy does the cell take to move through it? And the question that we're asking is really highly enabled by the field. So if you know anything about metabolism, the most classic way to study metabolism is a platform called Seahorse. And basically what Seahorse does is it tells you about basically the oxygen consumption of tens of thousands of cells at a time. So what's the oxygen consumption of this population of cells? Well, that's not really going to get at our question. We don't want to know about the metabolism of a population of cells. We want to know about the metabolism of a single cell as it's migrating. So we want to be able to correlate the single cell behaviors with the energy uptake. And so our work really started with when these biosensors came out. There's a number of them on the market now. This particular one measures the ratio of ATP to ADP. So it basically tells you how much active energy the cell has at a given time in the form of ATP. There's now a lot of these biosensors. You can get them for oxygen or pyruvate or NAD+, basically name your favorite metabolic marker. And the advantage of it is that it allows you to look at a single cell, identify what its energetic state is, and then correlate it with the behavior in real time. So when these sensors came out, they actually started using them in bacteria mostly, a little bit in neurons. We were the first to put them into cancer cells. And so you can see this here. This is an MDA MB231 cell. This is a breast cancer, highly metastatic breast cancer cell embedded in collagen. It's expressing the ATP probe. In a separate channel, you can see the ADP probe. You divide one by the other and you get what they call the Percival HR signal. So it's basically the ratio of ATP to ADP. Since we were the first to put in these cell types, we had to check and make sure it works. So we basically gave the cells increasing amounts of glucose. So this is giving them more and more fuel. And what we find is they produce more and more energy. So their ATP to ADP ratio is going up. Likewise, if we give them serum, we see that ratio go up. Okay, so back to the question. So how does migration correlate with energy generation? So these were pretty painful experiments that my graduate student did. But basically what he did was he monitored cells over time, taking images every minute of their speed and of their ATP to ADP ratio. And so this is just one trace example. So this is a cell. This is its ATP or ADP ratio. You can see it bounces up and down, and it actually bounces up and down with the speed. And we calculated that. We did a correlation analysis and found that they're actually highly correlated, speed and ATP burn, with about a 2-minute lag. So basically, cells will burn ATP, and within 2 minutes, their speed goes up. So this was really highly correlative, right? It's all based on correlation, but the first evidence that ATP levels are going to change due to migration behaviors. Okay, so if that's the case, if we make it harder for the cell to move, if we make the environment denser and requiring basically more cell activity, presumably it needs more energy. And so we wanted to test this. So we put cells into collagen of increasing density, and then we measured the ATP to ADP ratio. And so you can see here when the matrix density goes up, the ATP to ADP also goes up. So this was really interesting. This is what we thought would happen. But then somebody asked us an intriguing question, which is, how do you know that the energy is going up because the cells need more? Maybe the energy is going up because the cells aren't utilizing as much energy. So they're not using as much, and so they have more there, and that's what we're measuring. So we did a few other experiments. The first was we used a probe called 2-MBDG. This is a glucose analog. It's fluorescent. As the cells pick it up, they glow, and the more they pick up, it's indicative of increased glucose uptake. So basically, it's the input. And what we see is that goes up with collagen density. So the cells are taking in more glucose as the density of the collagen goes up. And not only that, we also looked at ATP hydrolysis, so the burn of ATP, and that also goes up. So as you make the environment harder to move through, cells take in more glucose, they make more ATP, and they burn more ATP. So they basically become more metabolically active. Okay, but what if we make it easier for the cells to move now? So what we did here was we created aligned collagen matrices, and these are well known to affect migration. We didn't discover this at all. It's been well established that if cells are migrating on aligned collagen, they increase their speed, which you can see here, and they tend to be more motile. So it's basically like laying down highways for them. If you can lay down highways, then they move much more easily. So we made aligned collagen. This is just us quantifying it here, saying our pixels, our collagen fibers are well aligned. Our cells move faster. And then if you look at all the same metrics we just looked at, cells require less ATP, they pick up less glucose, and they burn less ATP when you put them in easier to move matrices. So this is, again, showing us that migration is an important factor in determining metabolic activity and vice versa. Okay, so back to our questions. What about energy affecting migration decisions? So given that metabolism is going to affect migration, do cells actually choose how they're going to migrate based on how much energy they have? We did something somewhat clever here that we had been using this technology for a while of basically trying to recreate the microtracts that exist in vivo. So again, these are images from a tumor. You can see there's a lot of interstitial spaces that exist between the collagen. And we developed a platform to basically recreate these. And it's basically based on micromolding. We create a PDMS stamp and press it into collagen as it's polymerizing. So it's a lot like working with Play-Doh. And then we seed it with cells and we cap it. And so what you get is basically a cell that's fully enclosed in collagen and adherent on all sides. And here's just a video so you can see that we basically have our cells running a race now in this nice aligned collagen track. Okay, so what this allows us to do then is try and create spaces that are increasingly difficult for the cell to move in, and then ask what kind of choices does it make in these, what kind of choices do they make in these cells, in these environments? So we started with a taper track. This is our engineering hat on, trying to develop boundary conditions of confinement. So how big should we make these channels? And we wanted to know first, at what size channel does the cell touch all 4 walls? So how big can we make it where the cell is still confined? And how small can it be before the cell turns around? So we basically made these tapered microtracks. We watched cells march down the taper. We marked the point at which they touch all 4 walls, and then we marked the point at which they turn around. And so this is this is a cell. It's on 1 wall. As it marches down our taper, you can see it connects with 2 walls. And then at about 7 microns, it turns around. So 7 microns is sort of the magical number where the cells aren't happy anymore and don't want to go in anymore. So what did we do with that information? We basically built a maze for the cells. So it's a cross street. The cells march down the main channel, they hit the intersection, and they have a choice between a 12 micron track, which is the track at which the cells are going to be contacting all 4 walls, or they have a choice for a 7 micron track. So this is right at the point where the cells aren't so happy and don't want to squeeze. And so what did we notice? First of all, when the cells are in this feeder track, this 15 micron track, they actually pull inward on the track. So it's so large that they're actually pulling the collagen in. For the 12 micron track, when they move through it, they are confined and they're pushing it outward. And when they're in the 7 micron track, they're really confined and really pushing it out. So basically, this is telling us that the cell is squeezing in the 7 micron track. So this graph's a little bit confusing, but let me walk you through it. So basically what we did was we first looked at cells that were in this feeder track and attached to the wall that leads into the narrow region. And that's this light blue bar here. So if the cell is on the wall that leads into the narrow region, it's most likely to enter that narrow region. If the cell, and less likely to enter the wide branch and not very likely to turn around. If the cell, on the other hand, is attached to this side wall that enters the wide branch, it's more likely to enter the wide branch. So that's this darker blue bar. What's the really interesting data, though, is the orange. So if a cell marches down this channel attached to both sides, it gets to the intersection, it sends up extensions into both sections, and then it's most likely to choose the wide branch. So if you give it a choice between a wide branch, a narrow branch, or turning around, it tends to choose the wide branch. So this was our first evidence that the cells are not agnostic to cell size, to channel size, that they're making a choice. Okay, so the hypothesis was that it's all energy based. So we simply looked at the amount of energy it takes to move through these channels. And as it turns out, in fact, it does take a lot more energy to move through a 7 micron track than it does a 15. And you can see that here. So significantly higher ATP in our narrow branch where the cells are squeezing. What's even more interesting, though, is that if you look at the energy levels far before the cells ever get to the branch, you can predict the likelihood that a cell will enter the narrow branch based on the amount of energy it had as it moved down that channel. So basically what we did was we tracked the ATP to ADP ratio as it moved down the channel, and then we tracked to see which direction it went when it hit the bifurcation. So the bifurcation occurs right here. This is the ATP to ADP levels as it hits the bifurcation. And what we find is that the cells that had more energy leading up to the bifurcation are more likely to choose the narrow path. So if a cell is higher energy, it's going to care less about which path it chooses because it has the energy to do so. Okay. So we did a lot more in that space. We manipulated cell stiffness, we manipulated matrix stiffness, we created a mechanical model to look at how the cells move. And basically everything pointed to the same thing, which is cells will make migration decisions based on how hard it is to move through a certain path. And in fact, cells are pretty lazy. They choose the easiest path and do so somewhat intelligently. Okay, so I'm going to switch gears just a little bit and talk about the role of energetics and leader-follower dynamics. So what I haven't introduced yet, if you work in the cell migration field, you're very aware of this, but cell migration for probably 50 years has been focused on individual cells. How do individual cells move? And in the past 5 years or so, the field is taking a shift and focusing more on collective movements. And so if you look at basically general mechanisms of cell metastasis, you can think about an individual cell breaking free from a tumor, moving out into the circulation. This does happen, but what they see actually more clinically, if you actually look at pathology slides, is collective invasion. So they don't move as one individual cell, they move as these clumps of cells with a leader cell out front and a bunch of follower cells. So the cell migration field is really heavily invested right now in understanding the mechanisms of collective migration. And so we wanted to look at the same thing. And our hypothesis here was that for collective migration to work, that this leader cell takes the brunt of the energetic needs. And the idea being that if you were to ask the question, what makes a leader cell a good leader? It's going to be ability to generate a lot of energy. because this leader cell is the cell that's carving the path for the cells behind it. So we had to create a system to study this. We used tumor spheroids. So this is basically clumps of tumor cells that we aggregate and embed in collagen. I can show you what they look like here. So they'll invade out, and there's our leader cell leading the collective migration behind. And we've done it with a number of different cell types. This is a breast cancer cell line. This is also a breast cancer cell line. This is a prostate cancer cell line. But you can form these collective strands in all of these cases. So what we did was we embedded them in collagen. And the first thing we noticed is that the leader cell does in fact do a lot of remodeling. So you can see it here. So we can zoom in. You can see that it actually bores tunnels in the collagen that the follower cells follow behind. It also does a lot of collagen alignment, which we've quantified here. So the collagen starts off as completely random, but these leader cells can actually align it, providing these tracks for the follower cells to follow behind. So again, this is well known. We just showed it in our system. And then we asked, what about the energetic needs of these cells as they move? What's interesting is if you watch these collective movements, what you actually see is that the leader cells will actually trade off. And so you can see this here. This is basically a time course. This is our follower cell. And you basically get to a point, follower cell takes the lead and the leader cell drops back. So our hypothesis here was that given that we think the leader cell is highly energetically demanding or has high energetic demands, the hypothesis is that as the leader cell depletes its energy stores, it drops back and allows the follower to take over. So how did we show this? We started by first looking at the glucose uptake of these leader cells. And in fact, the leader cells do take up significantly more glucose than the follower cells. So you can see this here. This red and pink is indicative of higher glucose levels than the follower cells. So our first hint that they're more metabolically active, they're processing more glucose. What's also interesting is that the lifetime of these leader cells is dependent on the matrix. So if you make the matrix harder to get through, the leaders tend to drop off and become followers even quicker. And so this is this analysis here. It's a Kaplan-Meier analysis. People use it for patient survival. We used it for leader cell survival here, and basically showed that if you make the matrix really hard to move through, 6 mg per mil, the leader cell drops off really fast and gets replaced by a follower cell. If the matrix is easier to move through, 1.5 mg per mil, then the leader cell lasts even longer. Okay, so if we now do that same analysis, but we now vary the density of the collagen and we look at leader cell glucose uptake, what we see is that the leader cell glucose uptake increases with the collagen density. So the harder it is to move through, the more energy the cells, the more glucose the cells are gonna uptake. What's interesting though is that the follower cells, regardless of the density of collagen, don't change their glucose uptake. So the leader cell is doing all of the work, and the follower cells, regardless of the environment, as long as the leader cell is doing the work, the follower cells don't really change. Their glucose levels stay flat. Okay, so again, there's a lot more to the story. We have a model of energetic utilization here, trying to understand how much energy it actually takes and trying to predict the timing of switching and things like that. But really the conclusion that we came up to is that cancer cells use drafting. So basically the same idea that geese use, you've probably seen it, this lead goose, he'll fly in the V formation. This helps cut down wind resistance. He's bearing the biggest burden of the flight. When he gets tired, he'll switch places and a follower will take over. And these geese will actually even honk at each other to say, you know, time to take over when they get tired. The same thing happens in a peloton of bikes. You guys are probably familiar with this if you know anything about cycling or even racing. The idea that this first, the car or bike in the lead is basically cutting through the wind, and the riders behind benefit from it. And so we think cancer cells basically use the same mechanism. And we have a lot of questions that are coming from this now. So how do they communicate with each other to make the switching? Is it passive or is it actually active? And how do they determine at what point they actually drop back? Again, one of the things that I promised at the front end of this is not just the research for the sake of doing research, but what does this mean actually in translating it? Because we know migration is energy intensive, it's energy intensive like proliferation is. We know that leader cells are playing this really important role in cutting through the matrix. We know that now that metabolism, the migration, and the extracellular matrix are linked. The reason we're excited about this is because in the field, there's a number of metabolic inhibitors that are now working their way through the FDA pipeline. And they're working their way through looking for effects on proliferation and primary tumor growth. And one of the things that we're trying to say is, what about using these drugs to inhibit migration as well? And currently, there are no drugs on the market targeting metastasis or metastatic migration. And so we're asking whether or not these drugs can get repurposed. And again, the reason we're excited, the first metabolic inhibitor came out in the market just a couple of years ago, 2017. It wouldn't be good for migration. It's mostly targeting immune cell metabolism. But nonetheless, it's paving the way for a lot of different metabolic inhibitors coming out. So I'm going to switch gears a little bit to the second half. I promised I would talk about two different things. The first was metastatic migration. The second I'm going to talk about is some work we've been doing in angiogenesis. For graduate students who might be sitting there and thinking, how am I? Where's my work going to lead? Where am I going to be 15 years from now? Where's my PhD going to go? If I go into academia, what else am I going to do with it? This actually is the longest running project in my lab. It's something that I was thinking about as a graduate student. When I was a graduate student, I was thinking about angiogenesis in the context of wound healing and how endothelial cells assemble during wound healing. And here I am now, 20 years later. So I can tell you where we've come in that time. And so we've taken some of those concepts of as you probably all know, one of the greatest limitations to tissue engineering is coming up with a blood supply, developing patent blood vessels. That's where we used to be research-wise in my head. And now I've translated some of the exact same ideas that we were looking at there to understand what goes wrong during tumor angiogenesis. And a lot of this work was inspired by Rakesh Jain. He's an engineer at Harvard. And what he has noticed is that while tumor angiogenesis is known to occur, we know that for tumors to grow, they need a blood supply. What happens though is that the blood vessels that form hardly look normal. So they tend to be very tortuous and leaky. So this is normal blood vessels. These are tumor blood vessels. They tend to be very branched, tortuous, leaky. The problem with these blood vessels is that they then have several issues. They have trouble feeding the tumor properly. The tumor can actually undergo hypoxic conditions due to lack of nutrients. And unlike the convention would tell you, oh, well, maybe the tumors will starve. In fact, oftentimes what happens is the tumors get more aggressive. And so Rakesh has really been pioneering this idea of trying to actually restore the integrity of these blood vessels so that you don't get these high interstitial pressures, you don't get this necrosis, you don't get hypoxia, but you can actually use the blood vessels as highways to get drugs delivered into the tumors. And so the field, though, in terms of this, has caught on to it, but is almost exclusively interested in growth factor receptors. So can we come up with inhibitors against VEGF or VEGF receptor? One of the biggest drugs on the market that was just pulled a couple of years ago, Avastin, went after the VEGF receptor. So really the focus in the field has been on the receptors and the growth factors. But we're engineers, and so we've been thinking about it more from a mechanical standpoint, asking the simple question, given that solid tumors are stiffer than normal tissue and the tumor vasculature is abnormal, whether or not the matrix stiffness contributes to the abnormal vascular structure. And really the work was inspired. This was one of the very first papers. It was the very first paper I ever published when I started in my lab back in 2008. And it was we simply noticed that if you take cells, you put them on soft substrates, they tend to self-assemble and form these nice tight connections. If you take endothelial cells and you put them on stiff substrates, they just spread. There's no special connectivity there. So basically, by simply being in a compliant environment, you can induce cells to self-assemble into these networks. So what we noticed, if you look at the field actually, is that everybody who does in vitro angiogenesis assays uses these fairly soft substrates. Fibrin, which is actually not all that physiological, matrigel, again, not very physiological, and collagen. And if you just take endothelial cells, you put them into these soft environments, they'll self-assemble into these network structures. So each of these has a very different chemical composition, and yet you can get them to self-assemble. And the commonality between them is that they're all soft. So our question was whether or not the compliance, how soft the material is, whether or not that was affecting vessel formation. So one thing that we had to do was we had to create 3D matrices where we can control the stiffness. For this, we used collagen, and we used a naturally occurring process called glycation. So glycation is basically when sugars cross-link proteins. So you can take a collagen matrix, you can soak it in high concentrations of sugars, and you can get materials that are increasing in their stiffness. So you can get really soft matrices, about 300 pascals, and depending on the density of the matrix, we went up to about 750 here. You can go up to a couple thousand by increasing the density. Okay, so what we noticed is we took these matrices, we put in endothelial cell spheroids, and we simply looked at outgrowth. And what we found is that if you increase the stiffness of the matrix, so this is fairly soft, stiffer, and even stiffer, you can increase the outgrowth, the rate of angiogenesis. And so this is quantified here. You get significantly longer extensions and you get significantly more by simply making the matrix cross-linked and stiff. These are just prettier pictures of what I just showed you of these individual spheroids, but what you can see really clearly is significantly more outgrowth over time, and not just more outgrowth, but more branching. So remember I told you one of the hallmarks of tumor angiogenesis is tortuosity and branching, and we get significantly more branching just by increasing the stiffness. And we've quantified that here. So you can see the branch density goes up with the stiffness of the matrix. We wanted to move into a slightly more physiological system. So we used a chick assay. This is a really common assay in angiogenesis. Basically, it's a fertilized egg that we crack into a hammock. It's basically a piece of saran wrap, and you keep it incubated. And you can watch the blood vessels develop in real time. So we took a collagen matrix, we implanted it on top of these blood vessels, and then we just watched to see what their ingrowth looked like. And what we saw is that when you increase the stiffness, you increase the glycation of the matrix, you increase the vessel density of the vessels growing in. Okay, we also wanted to move in vivo. For this, we used a mouse model. So this is a MMTV PYMT mouse. It spontaneously develops breast tumors. You can control the stiffness of those tumors by giving the mice this chemical called BAPin. It basically prevents lysyl oxidase activity, which is a collagen crosslinker. So the tumors develop, they just develop much softer. And then we used Doppler to look at the vessel structure. And what we saw was that in our BAPin tumors that are much softer, we see significantly less vessels. So you can just compare this image to this image. You get significantly less vessels in our soft tumors compared to our stiff tumors. And we've quantified that here. So you get significantly more branching in stiff tumors and significantly more junctions. So basically that tortuous architecture. Okay, so I'm going to take it just full around. We talked at the beginning about metabolism. How does this all relate back? Well, one of the questions we're asking now is that increased branching that we see in our mice and in our in vitro systems, whether or not it's actually guided by metabolism. So in other words, do branches make the decision, do outgrowths make the decision to branch because the metabolic needs are too high to go forward and so that they're going to branch off? And in fact, when you increase the density of the collagen, you do increase the ATP to ADP ratio, the amount of energy the cells need to produce these angiogenic tips. So this is basically the direction we're headed now, which is correlating our angiogenic phenotype to our metabolic phenotype. Okay, so with that, I'm going to just summarize the second part of my talk, which is that matrix stiffening through changes in density and cross-linking can disrupt vascular growth and structure. And really, again, one of these goals is not just to discover for the sake of discovery, but also to try and relate it back to what we can do to inhibit this process. And this was a review we published a couple years ago. All credit goes to my student, Marshall Lampy, who's now on the faculty at the University of Houston, wrote a beautiful review, basically looking at all of the drugs that are currently in clinical trials that have potential for mechanobiology approaches and looking at things that can affect things like stiffness and force and contractility that might be purposed for mechanomedicine. And so with that, I'll acknowledge our funding, the NSF and the NIH. And the folks that did the work, Matt Zanatelli did a lot of the metabolic work that you see here, and Jan Zhang, a really talented postdoc in my lab, did all of the collective migration studies. And then the angiogenesis stuff was mostly done by a former postdoc, Frank Bordelot, who's now a faculty member up in Canada at Laval University. And with that, I will take any questions.
Wow, wonderful! I'm so happy and you know and be part of this this amazing talk. Many of us suffer of a friend or a family with cancer, and always metastases have been the main concern. So thank you very much for sharing those findings from your lab, and you know, this important tool that could be therapeutic in the future. We have many questions, so I will go quickly. And first, we are so lucky today. We have Dr. Koss, who is the associate dean for the University Graduate School, and Dr. Koss work on focus on cancer. So she have the first question for you. Can you comment on the timing of metastatic cells migration in relation to targeting it therapeutically?
Oh, that's I think I'm echoing or something. That's a great question. So, you know, I think one of the struggles clinically that they have is really predicting the timing of metastasis and really identifying if it's occurred yet. So clinically, basically what they do is they look to see the invasive front of a tumor. So if they remove a tumor, they look at the invasive front to see how far it's invaded. They look around at lymph nodes to see if they can find it in the lymph system. And oftentimes do scans to see if they can find metastatic cells in other sites. But in terms of the exact relationship between how fast cells migrate and how quickly they're likely to metastasize, I think that's actually one of the big questions in the field, is really trying to figure that out. And you can imagine how it would significantly change how you're going to deal with a patient if you know whether or not the tumor is likely to have metastasized, or where the cells are. If you could figure out where every single cell had gone, or the likelihood that it's going to show up in a metastatic site, I think you'd be much more aggressive, for instance, with treatment. And in my understanding clinically is that that's actually one of the biggest hurdles that doctors would like to overcome.
Excellent. So we have 54 participants today in your talk. So I don't know if we will be able to cover all questions, but I have here one. I will go through the order from Valentina Dargan, one of our PhD students. Have you compared the ratio of ATP use versus ATP synthesis? Can this influence cell migration as well?
That's a good question. So I think that the question is the best way to look at ATP synthesis. So one of the reasons we use these probes is because they were one of the first available at the time that the biosensors weren't really coming out as quickly. Now we have a number. We haven't published with most of them yet because we're still trying to figure out how they work and calibrate them. The probes, I'd like to say they're easy, but they're actually a little bit tricky because it's not necessarily sort of black and white. And so the problem with looking at other things like synthesis or other metabolic players is really just making sure the probes work the way that you think they are. In terms of looking at synthesis, there's different probes for different stages of glycolysis and the oxidative phosphorylation. So I don't know that there's a probe that specifically tells you when ATP pops up, but there's probes, for instance, that tell you lactic acid production or pyruvate. So in theory, you could sort of build up the entire cycle and see where every molecule is coming into play and how quickly it's happening. We haven't done that yet, but again, in theory, if you had enough channels on your microscope, you could really get a readout of almost every stage of the metabolic cascade.
Excellent. So I will jump here and to a question from our chair, Dr. John. What triggers the initial leader cells?
That's a good question. So one of the biggest questions in the field right now is what makes a leader a leader? And I can tell you, we have some work we haven't published yet. It's actually pretty old. We've just sort of been sitting on it, but you can take, so for some of our collective migration experiments, we use like a ball of otherwise non-invasive cells. They're non-malignant, they're non-metastatic. And we create a ball of them, and we will put in a single metastatic cell. So you'll have 10,000 non-metastatic cells, one metastatic cell. And that metastatic cell, we can watch it bounce around in the spheroid, move to the edge of the spheroid, start migrating out, and all of the other cells will follow behind it. How it is that cell finds itself to the edge, we don't know if that's an active process or just accidental, just found its way on the end. But we can watch it as it starts to move. It will start to realign the collagen. It contracts all of the collagen around it and carves a path that the follower cells can follow. And so one of the questions people are asking is, what makes a leader a leader? So what are the properties that have been endowed in that cell that tell it to lead and trigger it to go? And that is not at all clear. I mean, I think part of it, my own bias is saying part of it is likely the extracellular matrix and the extracellular matrix cues. But other than that, what specifically triggers a cell is not clear.
Good. We continue here. We have a question from Daniel Chaparro. Can the energy used for motion be stored elastically in intracellular fibers?
Oh, that's a good question. So we have a paper now we've been wrestling with at JCB. It's in its second revision, looking at exactly that question. So the tension gets created in the matrix compared to the alignment in the matrix and the forces the cells exert. So what we do know, this is actually work Sanjay Kumar has produced. What we do know is that if you look at those fibers and you cut them, so he uses basically like a laser knife to cut individual fibers that the cells are pulling on, you can see the fibers spring back. So there is elastic energy in those fibers as the cells are pulling on it, for sure. And those cells can interpret that elasticity. So they view that elasticity, that tension, as a trigger for migration. So the short answer is yes, absolutely.
Okay, so Dr. Hutchinson, by the way, Dr. Hutchinson was the person who initiated this contact. As we mentioned at the beginning, we appreciate that. Dr. Hutchinson did really cool work using your patent collagen construct. Are you able to decouple the energetic associated with fiber density versus stiffness of the matrix?
Oh, so this question is one what we've been we are huge fans of and view as incredibly important. And I think there's a lot of folks that confound those two things and use them sort of interchangeably. And so we do our best not to do that because stiffness can come in two forms. It can come due to matrix density or it can come due to cross-linking. And so in our system, we keep trying to come up with new ways to cross-link the matrix to increase stiffness that don't affect cell binding, that don't affect density, so that we can decouple. It is definitely a tricky question. I think people that are better biochemists than matrix biochemists than I am are coming up with better materials all the time. But it's most definitely a question that we wrestle with.
Excellent. We have a question from an anonymous person. Would cancer cells give up? Could they run out of energy and stop trying to metastasize?
Oh, I wish. So I think, unfortunately, what we do know about cancer cells with regard to metabolism is that they are incredibly malleable and adaptable to their environment. And it's one of the things that makes them quite dangerous. So you can starve tumor cells of nutrients, you can deprive them of oxygen, you can create highly acidic environments, and there are cancer cells that will alter their metabolic pathways to get around that and find new nutrients to feed their metabolic needs. And so, yes, in theory, they deplete energy as they move, but they're also really good at rebuilding it despite whatever environment they're in.
We have another question from our chair, Dr. John. Could collagen alignment be monitored early to see if that is changing and then disrupt it?
Oh, that's a good question too. So there's some work by Jan Liphart's group that basically tries to blow up collagen fibers so that cells can't follow along. And that is basically what happens. So if cells go through all that effort of aligning collagen fibers and then you just completely blow them apart and you release that tension, you release the alignment, the cells don't follow it anymore. They basically stay put. The problem though in the field with that, if you're trying to think about like therapeutic options, is that there are a number of fields like atherosclerosis. We do a lot of work in athero where we know that matrix stiffening is playing a key role. Matrix stiffening happens in age, changes in alignment happen with age. There's a number of pharmaceutical companies that are hot on it. They know that that's the case, but the drugs to target it just continue to fail. That the ability to disrupt cross-linking and alignment and things like that. I certainly would not advocate for people to give up on that. I think that it's a very promising avenue, but I think it's been difficult to do.
We have another question from Danny Chaparro. Do you think a cell ability to deform a tight channel, for example, the 7 micrometer channel with differing stiffness, affect its past decision making? Yeah,
absolutely. So we know that I didn't get a chance to show this data, but if we take a 7 micron path and we cross-link the collagen, so we make it stiffer, the cell becomes even less likely to choose that path. So if we stiffen the cell, which is a tricky experiment to do, but if we do it, it tends to alter a lot of other things as well. But if you look at it just as a function of cell stiffness, the cell becomes less likely to choose the narrow path. So basically, cells are making choices on whether or not they have the energy to move through a path. And what's cool, again, I didn't get a chance to present it, but cells will send out extensions into each of these paths. They'll even put up mitochondria into those extensions and look at the energetic needs. And they don't just go, they sit at the bifurcation, they sense one, they sense another, and then they make a choice. And so clearly there's feedback that they're getting to make a decision on which direction they're going to go.
Excellent. I have one question for you. So this is not my area of research, but I'm wondering if stopping energy consumption on target cell, it could be a useful therapeutic in this case. Can we create some method that specifically target the hexokinase of the glycolysis in those cells that are leader cells, and then we stop? Is there any way we can just using antibodies or something, or knowing they are a specific subtype of that protein in the entrance of the glycolysis, the HK, can we do something at that respect?
I think so. So I think that that's where the field is going, which is targeted metabolic inhibitors. And I think there's 2 ways to do it. The first is to identify metabolic pathways that are uniquely upregulated in cancer and go after those. The second is targeted therapeutics. So basically figuring out how to deliver drugs that, in theory, if you got them anywhere, would be quite dangerous. But if you deliver them directly to tumors, are going to inhibit metabolic activity just there. And I think that holds a lot of promise. I think it's actually surprising to me that metabolic inhibitors haven't been more quote unquote popular before more recently. I mean, almost every drug goes after just the ability of a cell to divide. So it goes after like mitotic spindle assembly and things like this. The metabolic inhibitors, I think you're going to see over the next 5 to 10 years an explosion of metabolic inhibitors trying to inhibit metabolic pathways associated with tumor cells.
Okay, so I think we are 9:59, just on time, and I would like to thank again Dr. Reinhart-King for being here with us today, even in a busy day for her. Thank you very much. And I invite all the students and all the participants to join our poster session that will start at 10:30, and there will be breaking room for posters. So feel free to join any poster. I know that this is a new way to interact, and we invite you to enjoy the poster presentation and the other part of the program. Thank you so much, Dr. Reinhart-King, for being with us.
Thank you for having me, guys. Enjoy your day.
We have you here physically shortly, I guess.
Well, enjoy your gorgeous weather as well.
Thank you.